Welcome to the For Science Salah Lotfy channel, where we explore the fascinating frontiers of scientific research. Today we bring you a groundbreaking study that uncovers the hidden interplay between human cells and the microbiome in a severe and life-threatening condition, oral squamous cell carcinoma. Oral squamous cell carcinoma is one of the most common cancers of the oral cavity, affecting thousands of lives globally each year. This type of cancer originates in the squamous cells that line the surface of the mouth, tongue, and lips. It is particularly aggressive and often diagnosed at a late stage, which complicates treatment and reduces survival rates. Its survival rates remain alarmingly low, making it a critical focus for medical research. Scientists are tirelessly working to develop better diagnostic tools and more effective treatments to improve patient outcomes. While risk factors like tobacco and alcohol are well known, they are not the only contributors to this disease. Lifestyle choices and genetic predispositions also play significant roles. Recent studies have turned their attention to an unexpected player, the microbiome. Researchers are exploring how the balance of bacteria in our mouths could influence the development and progression of oral cancers, opening new avenues for prevention and treatment. This study employs advanced metatranscriptomic techniques, allowing researchers to explore the genetic material of entire microbial communities. By diving deeper than ever into the microbiome's functional role within tumors, scientists can uncover hidden interactions and mechanisms. The findings reveal a unique, multi-kingdom microbial community in cancerous tissues, including bacteria, fungi, and viruses, each playing distinct roles. Notably, certain microbes show active transcriptional profiles, indicating their dynamic presence and activity within the tumor microenvironment. This suggests their potential involvement in tumor development and progression, offering new insights into cancer biology and potential therapeutic targets. But this research goes beyond merely identifying microbes. It delves into understanding the complex interactions between these microorganisms and their human hosts. By integrating microbiome and host transcriptome data, it explores how specific microbial species might interact with human genes, revealing intricate relationships that could influence health and disease, potentially driving pathways linked to cancer proliferation. This insight opens new avenues for understanding how microbial communities might contribute to cancer development. For example, one bacterium was found to upregulate genes tied to cellular growth, highlighting the potential for microbes to influence cellular behavior in significant ways. A discovery validated through rigorous laboratory experiments, underscoring the importance of experimental validation in confirming these groundbreaking findings. The study also sheds light on field cancerization, a phenomenon where non-cancerous tissues adjacent to tumors exhibit early oncogenic changes, possibly influenced by the surrounding microbiome. Join us as we delve into this cutting-edge research that bridges microbiology, genomics, and oncology, offering fresh insights into cancer biology and future therapeutic approaches. Don't forget to subscribe and share this video to support our mission of making science accessible to all.
Welcome to the For Science Salah Lotfy channel. Today, we're excited to present a fascinating research study that explores the intricate relationship between the microbiome and oral squamous cell carcinoma, also known as OSCC. The research is titled, Integrative Metatranscriptomic Analysis Reveals Disease-Specific Microbiome Host Interactions in Oral Squamous Cell Carcinoma. This groundbreaking study was conducted by a distinguished team of researchers. First, we have Dr. Vinay Jain from the Bhabha Atomic Research Center in Mumbai, India, and the Maurice H. Kornberg School of Dentistry at Temple University in Philadelphia, USA. Next is Dr. Divyashri Barania from the Maurice H. Kornberg School of Dentistry, Temple University, Philadelphia, USA. Then Dr. Doa El Haddadi, also from the Maurice H. Kornberg School of Dentistry, Temple University, Philadelphia, USA. Dr. Tsute Chen from the Forsyth Institute in Cambridge, Massachusetts, USA. Dr. Michael Slifker from the Fox Chase Cancer Center in Philadelphia, USA. Dr. Fadil Alaqua from the University of Michigan, Ann Arbor, USA. Dr. Kathy Kai from the Fox Chase Cancer Center in Philadelphia, USA. Dr. Kumaraswamy Chitrala from the Lewis Katz School of Medicine at Temple University, Philadelphia, USA. Dr. Christopher Fundakowski from Thomas Jefferson University, Philadelphia, USA. And finally, the corresponding author, Dr. Nezer Al-Hebshi from the Maurice H. Kornberg School of Dentistry, Temple University, Philadelphia, USA. and the Fox Chase Cancer Center, Temple University Health System, Philadelphia, USA. This study was published in the prestigious journal Cancer Research Communications. Date of submission: October 3, 2022. Date of acceptance: April 13, 2023. Date of online publication: May 8, 2023. This research offers groundbreaking insights into the functional role of the microbiome in OSCC using cutting-edge techniques like laser microdissection and ultra-deep metatranscriptomic analysis. Stay tuned for future videos where we'll dive deeper into the findings and implications of this exceptional study. Thank you for watching. Don't forget to like, share, and subscribe to the For Science Salah Lotfy channel for more insights into the world of scientific discovery. Oral cancer is a formidable foe. It often remains undetected until its later stages, which makes treatment more challenging. Oral Squamous Cell Carcinoma, or OSCC, is the most common type. OSCC affects millions worldwide and claims countless lives each year. This silent threat often begins with subtle symptoms that can easily be overlooked. Early detection is crucial for successful treatment. However, many patients are diagnosed when the cancer has already spread. This highlights the urgent need for better understanding and earlier intervention. We must shed light on the complex factors driving OSCC. This knowledge will pave the way for innovative prevention and treatment strategies. We are not alone. Trillions of microorganisms inhabit our bodies. This vast community is known as the microbiome, and it plays a crucial role in our health. The microbiome is a complex ecosystem consisting of bacteria, fungi, viruses, and other microbes. These microbes outnumber our own cells. Recent research has revealed a fascinating link. The microbiome may play a role in cancer development, including oral cancer. Understanding this intricate relationship could revolutionize our approach. We may be able to develop new ways to prevent and treat OSCC. Unraveling the microbiome's role in OSCC is no easy feat. Scientists are using cutting-edge techniques to investigate. One such technique is metatranscriptomics. Metatranscriptomics allows us to analyze the RNA of an entire microbial community, providing valuable insights into gene expression. We can identify which genes are active in the tumor microenvironment. Another powerful tool is laser microdissection. This technique allows researchers to isolate specific cells or regions within a tissue sample, enabling the study of the microbiome in very specific locations. These advanced techniques are providing unprecedented detail, and we are gaining a deeper understanding of the microbiome's role in OSCC. Recent studies have uncovered fascinating findings. They highlight the microbiome's potential influence on OSCC. These discoveries have significant implications for future research and treatment. One key finding is the altered microbial composition in OSCC patients. Studies have shown distinct differences in the oral microbiome between healthy individuals and those with OSCC. These alterations in the microbiome may contribute to tumor development and could also influence the progression of the disease. Understanding these changes is crucial for developing targeted therapies. Researchers are also investigating the role of specific microbial species. Some species may promote tumor growth, while others may have protective effects. Section 5. Cutibacterium acnes, a closer look at a key player. Among the many microbial inhabitants of the oral cavity, one bacterium has emerged as a potential key player in OSCC, Cutibacterium acnes. This bacterium, which is more commonly known for its role in causing acne, has recently been identified in the oral microbiome. Researchers are now investigating its potential involvement in the development and progression of oral squamous cell carcinoma, OSCC. This bacterium, often associated with acne, has been found to exhibit intriguing behavior in the context of oral cancer. While it is typically found on the skin, its presence in the oral cavity has raised questions about its role in oral health and disease. Scientists are particularly interested in how Cutibacterium acnes might influence the tumor microenvironment. Studies have revealed that certain enzymes produced by Cutibacterium acnes are present at higher levels in OSCC tumors compared to healthy tissue. These enzymes, which are known to break down various molecules, could be altering the environment around the tumor, making it more conducive to cancer growth. These enzymes, capable of breaking down specific molecules, may play a role in modifying the tumor microenvironment. By breaking down these molecules, The enzymes could be facilitating the spread of cancer cells, thereby contributing to the aggressiveness of the tumor. The increased activity of these enzymes could potentially contribute to tumor growth and spread. This suggests that Cutibacterium acnes might not just be a bystander in the oral cavity, but could be actively involved in the progression of OSCC. Further research is needed to fully elucidate the role of Cutibacterium acnes and its enzymes in OSCC development and progression. Understanding these mechanisms is crucial for developing new therapeutic strategies that target the bacterium and its enzymatic activity. Understanding the mechanisms by which this bacterium interacts with the tumor microenvironment could open up new avenues for targeted therapies. By targeting the specific enzymes produced by Cutibacterium acnes, it may be possible to develop treatments that inhibit tumor growth and improve patient outcomes. Section 6. Field cancerization, beyond the tumor's edge. This section delves into the intricate and often overlooked aspects of cancer development, particularly focusing on the surrounding environment of the tumor. The concept of field cancerization adds another layer of complexity to our understanding of OSCC. It challenges the traditional view that only the tumor itself is the problem, highlighting the importance of the surrounding tissue. This concept suggests that the tissue surrounding a tumor is not merely passive but undergoes changes that make it more susceptible to cancer development. These changes can be subtle and not immediately visible, yet they play a crucial role in the progression of the disease. Imagine a field of grass where a fire has erupted. The fire represents the tumor, but the story doesn't end there. While the flames are concentrated in one area, the surrounding grass is also affected, becoming drier and more easily ignited. This analogy helps us understand how the tissue around a tumor can become a breeding ground for further cancerous changes. Similarly, in OSCC, the apparently healthy tissue adjacent to the tumor can harbor molecular and cellular alterations that create a fertile ground for cancer cells to thrive. These alterations can include genetic mutations, changes in cellular behavior, and even shifts in the local microenvironment. This field of precancerous changes can extend beyond the visible tumor margins, making complete surgical removal challenging. Surgeons must be aware of these hidden dangers to ensure they remove as much of the affected tissue as possible. Understanding the factors contributing to field cancerization, including the role of the microbiome, is crucial for developing effective treatment and prevention strategies. The microbiome, which consists of the microorganisms living in our bodies, can influence cancer development in ways we are only beginning to understand. By targeting the entire field of precancerous changes, we may be able to prevent or delay the recurrence of OSCC. This holistic approach could revolutionize how we treat and manage cancer, offering hope for better outcomes and longer survival rates. Section 7. The dance of microbes and host, a complex interplay. This section delves into the fascinating and intricate relationship between the oral microbiome and its human host. The relationship between the oral microbiome and the host is not a one-way street. Instead, it is a complex, bidirectional interaction where both the microbiome and the host exert influence on each other. It's a dynamic interplay, a delicate dance where both partners influence each other. This dance is continuous and ever-changing, reflecting the constant state of flux within our bodies. Changes in the host, such as diet, stress, or overall health, can impact the composition and function of the microbiome. For instance, a diet rich in fruits and vegetables can promote a healthy microbiome, while stress can have the opposite effect. Conversely, alterations in the microbiome can influence the host's immune response, inflammation levels, and even cellular processes. This means that the microbiome can play a role in either promoting health or contributing to disease. This intricate communication occurs through various mechanisms, including the release of signaling molecules and metabolites. These chemical messengers help coordinate the activities of both the microbiome and the host. In the context of OSCC, this interplay becomes even more critical. The balance between the microbiome and the host can significantly affect the progression of the disease. Dysbiosis, an imbalance in the microbiome, can disrupt the delicate equilibrium, potentially creating an environment that favors tumor development and progression. This imbalance can be triggered by various factors, including poor oral hygiene and systemic health issues. Understanding the intricacies of this microbial-host interaction is essential for developing personalized approaches to OSCC prevention and treatment. By gaining deeper insights into this relationship, we can create more effective strategies to combat this serious condition. Section 8, Shaping the Future, From Research to Therapy. The discovery of the microbiome's potential role in OSCC has opened up exciting new avenues for research and therapy. Scientists are exploring innovative approaches to manipulate the microbiome for therapeutic benefit. One promising area is the development of targeted probiotics. These beneficial bacteria could be administered to restore a healthy balance within the oral microbiome, potentially inhibiting tumor growth or enhancing the effectiveness of conventional treatments. Another approach involves targeting the metabolic pathways of specific microbial species. By inhibiting enzymes involved in tumor-promoting processes or promoting the production of beneficial metabolites, we may be able to modulate the tumor microenvironment. Furthermore, understanding the microbiome's role in field cancerization could lead to novel strategies for preventing OSCC recurrence. Section 9. A call to action, the importance of early detection and prevention. While research continues to unravel the complexities of the microbiome's role in OSCC, one message remains clear. Early detection and prevention are paramount. Regular dental checkups are crucial for identifying any suspicious lesions or abnormalities in their early stages. Lifestyle modifications also play a vital role in reducing OSCC risk. Avoiding tobacco use, limiting alcohol consumption, and maintaining a healthy diet and oral hygiene practices can significantly impact oral health. By raising awareness about OSCC, promoting early detection, and adopting healthy lifestyle choices, we can collectively combat this silent threat and improve oral health outcomes worldwide. Remember, knowledge is power, and taking proactive steps towards prevention can make all the difference. Hey there, science fans! Welcome to another exciting journey into the world of science and discovery. Did you know there's a whole universe living inside your mouth right now? Yes, you heard that right. It's a bustling metropolis of microorganisms. It's true, we're talking about the microbiome, a bustling community of bacteria and other microorganisms that play a crucial role in our health. What's even more fascinating is that scientists are discovering links between the microbiome and diseases like Oral Squamous Cell Carcinoma, or OSCC, a type of mouth cancer. This connection is opening new doors in medical research. OSCC is a serious condition. It affects the tissues in your mouth and can be life-threatening if not detected early. Understanding it better is crucial. But here's the exciting part. Understanding the microbiome's role in OSCC could lead to new ways to diagnose and treat this disease. Imagine the possibilities. Stay tuned, because we're about to dive into a groundbreaking study that's changing the way we think about oral cancer. This research could revolutionize our approach to treatment. This isn't science fiction, folks. It's the future of medicine, and it all starts with tiny organisms you can't even see. The future is now, and it's microscopic. Now, before we get into the nitty-gritty of this amazing study, let's talk about how scientists make sure their research is top-notch. First things first, any study involving humans needs ethical approval. This means a group of experts reviews the study plan to ensure it's safe and ethical for everyone involved. This study got the green light, and for good reason. The scientists designed it meticulously, carefully planning each step to answer their questions about the microbiome and OSCC. They also wanted to make sure the results were reliable and could help other scientists in the future. It's like building a house. A strong foundation is crucial for a sturdy structure. All right, science detectives, it's time to collect some evidence. In this study, the scientists needed samples from two groups, patients diagnosed with OSCC and healthy volunteers. They took tissue samples from the OSCC patients' tumors and compared them to similar tissues from the healthy volunteers. Why two groups? Well, it's like comparing apples and oranges. By looking at the differences between the microbiome in healthy mouths and those with OSCC, scientists can start to pinpoint which microbes might be involved in the disease. It's all about finding clues. Section 4. Precision is key. Laser microdissection in action. Now imagine trying to study a single grain of sand on a beach. It's nearly impossible to focus on just one grain without the right tools. That's kind of what it's like to study specific cells within a tissue sample. The complexity and density of the tissue make it a daunting task. But don't worry, scientists have a cool tool for that: laser microdissection. This advanced technology makes the impossible possible. This technique uses a laser, yes, a laser, to separate the specific cells they need from the rest of the tissue sample. It's like magic in the lab. It's like using a tiny pair of scissors to carefully cut out the pieces of a puzzle you want to focus on. Pretty neat, right? This precision is unmatched. This level of precision is crucial for getting accurate results and understanding exactly which microbes are hanging out in the tumor cells. It opens up new possibilities in research and diagnostics. Section 5. Unlocking Genetic Secrets, DNA, RNA, and the Future of Cancer Research. Okay, we've got our samples and we've zoomed in on the good stuff. These samples hold the key to understanding the intricate details of cellular functions. Now what? Time to unlock the secrets hidden within the cells. This is where the real detective work begins. Scientists use special techniques to extract DNA and RNA from the samples. These techniques are precise and require a lot of skill and patience. Think of DNA as the blueprint of a cell, containing all the instructions needed for building and maintaining an organism, and RNA as the messenger carrying out the instructions. It translates the DNA's instructions into actions within the cell. By analyzing the DNA and RNA, scientists can identify which genes are turned on or off in the tumor cells compared to the healthy cells. This comparison is crucial for understanding cancer. This can reveal clues about how the microbiome might be influencing the development of OSCC. The microbiome's role is a new frontier in cancer research. It's like cracking a code that could lead to new ways to diagnose, treat, and maybe even prevent oral cancer in the future. The potential is enormous. This research is just the beginning. Each discovery opens up new questions and possibilities. The more we learn about the microbiome and its role in diseases like OSCC, the closer we get to finding new and innovative ways to keep people healthy. This knowledge could revolutionize medicine, so stay curious, keep exploring, and never stop asking questions. The future of science depends on inquisitive minds like yours. Our bodies are incredible universes home to trillions of microorganisms. This is the microbiome, a complex ecosystem that plays a crucial role in our health. We are just beginning to understand this hidden world which influences everything from digestion to immunity. Scientists are discovering fascinating links between the microbiome and various diseases, including cancer. These discoveries are reshaping our understanding of disease mechanisms. Oral cancer, particularly oral squamous cell carcinoma, or OSCC, is a significant health concern. It affects thousands of people worldwide each year. It is a complex disease influenced by many factors, including genetics, lifestyle, and now, the microbiome. New research suggests that the microbiome plays a critical role in the development and progression of OSCC. This groundbreaking study provides a deeper understanding of the intricate relationship between the microbiome and OSCC, revealing potential biomarkers for early detection. It opens up exciting possibilities for future therapies and early detection strategies, which could significantly improve patient outcomes. This is a significant leap forward in our fight against cancer, offering hope for more effective treatments. By exploring the universe within, we are uncovering secrets that could revolutionize healthcare, making it more personalized and precise. Join us as we delve into the fascinating findings of this study, which could change the way we approach cancer treatment. Discover the potential of the microbiome in the fight against OSCC and how it could pave the way for new, innovative therapies. Imagine trying to understand a conversation in a language you don't know. It's confusing, right? You might catch a word here and there, but the overall meaning remains elusive. That's what studying the microbiome was like before high-throughput sequencing. Researchers were like detectives with only a few clues, trying to piece together a complex puzzle. This revolutionary technology allows us to read the genetic information of trillions of microorganisms simultaneously. It's like having a magnifying glass that reveals hidden details. It's like having a universal translator for the language of life, enabling us to understand the intricate conversations happening within our bodies. In this groundbreaking study, researchers used cutting-edge sequencing technology to analyze samples from OSCC patients. They aimed to uncover the hidden secrets of the disease. They sequenced both the DNA and RNA of the microorganisms, as well as the RNA of the host cells. This comprehensive approach provided a holistic view of the interactions at play. The amount of data generated was astronomical. It was like trying to read every book in a massive library all at once. It's like comparing the information content of a library to a single book. The depth and breadth of data were unparalleled. This massive dataset provided an unprecedented view of the oral microbiome in OSCC. It was like seeing a detailed map of a previously uncharted territory. It allowed researchers to identify specific changes in the composition and activity of the microbiome associated with the disease. These insights were crucial for understanding the disease's progression. This information is crucial for developing targeted therapies and early detection methods. By decoding the language of life, we can pave the way for more effective treatments and better patient outcomes. Think of your body as a grand orchestra. Each cell plays a specific instrument, contributing to the overall harmony. Cancer disrupts this harmony, causing some instruments to play out of tune or too loudly. This disrupts the entire orchestra. The study looked at the host transcriptome, which is like the musical score for the orchestra. It tells us which genes are turned on or off in the host cells. By comparing the transcriptome of healthy and cancerous tissues, researchers can identify the genes and pathways affected by the disease. The study found significant differences in the expression of genes involved in cell growth, inflammation, and the immune response. Some pathways were upregulated, meaning they were more active in cancer cells. Others were downregulated, meaning they were less active. These findings provide valuable clues about how OSCC develops and progresses. Imagine a thriving ecosystem teeming with diverse life forms. Now, picture a sudden shift where some species flourish while others dwindle. This is what happens to the oral microbiome in OSCC. The balance of microbial communities is disrupted, creating a favorable environment for tumor growth. The study revealed a significant shift in the composition of the oral microbiome in OSCC patients. Some bacterial species, like Cutibacterium acnes, were found in much higher abundance in tumor tissues. Others, which normally keep harmful bacteria in check, were significantly reduced. This imbalance in the microbial ecosystem can have far-reaching consequences. It can lead to chronic inflammation, a weakened immune response, and even promote the growth and spread of cancer cells. Understanding these changes is crucial for developing strategies to restore a healthy balance and prevent OSCC progression. Section 5. Cutibacterium acnes, a tiny player with a big impact on cancer. In the vast universe of the microbiome, Cutibacterium acnes is a tiny player, often associated with acne. This bacterium is a common resident of our skin, and while it is mostly known for its role in causing pimples, recent research has uncovered a much more significant role. However, this seemingly insignificant bacterium has emerged as a potential key player in OSCC, or Oral Squamous Cell Carcinoma. This type of cancer affects the cells lining the mouth and throat, and understanding its causes is crucial for developing better treatments. The study found a startling connection between Cutibacterium acnes and the activation of a powerful cancer-promoting gene called MIC. This gene is known for its role in regulating cell growth and division, and its overactivation can lead to cancer. MIC is like the conductor of the cellular orchestra, controlling the expression of many genes involved in cell growth and proliferation. When MIC is functioning normally, it ensures that cells grow and divide in a controlled manner. When MIC is overactive, it's like the conductor gone rogue, driving the orchestra into a frenzy of uncontrolled growth. This leads to the rapid and unchecked proliferation of cells, a hallmark of cancer. This is what happens in many cancers, including OSCC. The overactivation of MIC leads to the aggressive growth of cancer cells, making the disease difficult to control. The study's findings suggest that Cutibacterium acnes may contribute to OSCC development by promoting MIC activation. This means that the bacteria could be indirectly driving the cancer's growth. This discovery opens up exciting new avenues for therapeutic intervention. By understanding the role of Cutibacterium acnes in cancer, researchers can develop new strategies to target this bacterium and its interactions with MIC. Targeting Cutibacterium acnes or its interactions with MIC could potentially halt or even reverse tumor growth. This could lead to more effective treatments for OSCC and possibly other cancers influenced by this bacterium. Section 6. The future of treatment. Harnessing the microbiome for cancer therapies. The discovery of the microbiome's role in OSCC opens up exciting new frontiers in cancer treatment. Imagine a future where we can manipulate the microbiome to fight cancer. This is the promise of microbiome-based therapies. One approach could involve using probiotics, which are beneficial bacteria, to restore a healthy balance to the oral microbiome. Think of it as sending in reinforcements to fight the harmful bacteria and restore order. Another approach could target specific bacteria, like to block their cancer-promoting activities. Microbiome-based therapies offer several advantages over traditional cancer treatments. They are often less toxic, have fewer side effects, and may be more effective in preventing cancer recurrence. While still in the early stages of development, these therapies hold immense promise for the future of cancer care. Section 7. Early detection. Listening to the whispers of the microbiome. Early detection is crucial in the fight against cancer. The earlier we can detect cancer, the better the chances of successful treatment. The microbiome offers a promising new avenue for early cancer detection. Just like a change in the sound of an orchestra can indicate a problem with an instrument, changes in the composition of the microbiome can signal the early stages of disease. By analyzing the microbiome of individuals at risk of OSCC, we may be able to detect the disease before any symptoms appear. This could revolutionize how we screen for and diagnose OSCC. Imagine a simple mouthwash test that could detect the early signs of cancer. This would allow for earlier intervention and significantly improve patient outcomes. Section 8. The uncharted territories of microbiome research. The human microbiome is a vast and largely unexplored frontier. We are only just beginning to understand the complex interactions between the trillions of microorganisms that call our bodies home. Like early explorers charting new lands, scientists are constantly making groundbreaking discoveries in the field of microbiome research. Each new study brings us closer to understanding the role of the microbiome in health and disease. This knowledge is essential for developing new diagnostic tools, therapies, and preventive measures for a wide range of conditions, including OSCC. The future of medicine lies in unraveling the mysteries of the microbiome. By continuing to explore this uncharted territory, we can unlock the full potential of the microbiome to improve human health. Section 9. Conclusion. A new frontier in the fight against oral cancer. The study linking the microbiome to OSCC is a major breakthrough in cancer research. It has unveiled a hidden world within us that plays a critical role in the development and progression of this devastating disease. This discovery has opened up exciting new avenues for developing more effective and targeted therapies. Imagine a future where we can manipulate the microbiome to prevent and treat cancer. This may seem like science fiction, but it is rapidly becoming a reality. With continued research and innovation, we can harness the power of the microbiome to fight OSCC and other cancers. The fight against cancer is a global effort. By working together, we can translate these groundbreaking discoveries into life-saving treatments and improve the lives of millions affected by this disease. We share our lives with a universe of tiny organisms. This is the microbiome. It exists everywhere in our bodies, including our mouths. Scientists are learning that the microbiome plays a crucial role in our health. Now, a groundbreaking study has shed light on its connection to oral cancer. This research reveals that the microbiome is not just a passive bystander. It is an active player in the development of oral squamous cell carcinoma, the most common type of oral cancer. Imagine a bustling city teeming with life. That is the microbiome. Each microbe, a tiny resident with a specific role. This delicate balance can be disrupted. When it is, diseases like oral squamous cell carcinoma can arise. Understanding this complex interplay is vital to developing new treatments. This study takes us one step closer. This research marks a turning point in our understanding of oral squamous cell carcinoma. It challenges previous assumptions. We now know that the microbiome is not merely a background player, it is a driving force. This discovery opens up exciting new avenues for diagnosis and treatment. We are on the verge of a new era in cancer research. To explore this microscopic world, scientists used cutting-edge technology. One such tool is ultra-deep metatranscriptomic analysis. This technique allows us to eavesdrop on the conversations between microbes. It reveals which genes are active and how they interact with each other. This provides invaluable insights into the microbiome's role in disease. Another technique used was laser microdissection. This method isolates specific cells from tissue samples. It allows researchers to study the microbiome of cancer cells separately from surrounding healthy tissue. This level of precision is crucial for understanding how the microbiome differs in healthy and diseased states. These advanced techniques provide an unprecedented level of detail. They allow us to see the microbiome in action like never before. By combining these methods, scientists were able to paint a comprehensive picture of the microbiome's involvement in oral squamous cell carcinoma. The study's findings were nothing short of groundbreaking. They revealed that the microbiome is not a passive collection of microbes. It is a dynamic and active community. The microbes within the tumor microenvironment communicate with each other and with cancer cells. This communication influences tumor growth and progression. This discovery challenges the traditional view of cancer development. It suggests that targeting the microbiome could be a promising new approach to treatment. By manipulating the microbiome, we may be able to disrupt the communication pathways that fuel cancer growth. This paradigm shift has far-reaching implications. It opens up a whole new world of possibilities for cancer research. The microbiome is a complex and dynamic ecosystem. Understanding its role in oral squamous cell carcinoma is crucial for developing more effective treatments. The study also made a startling discovery. It found that the microbiome is altered not just in the tumor itself. The surrounding healthy tissue also shows significant changes. This phenomenon, known as field cancerization, suggests that the microbiome plays a role in creating an environment that is conducive to cancer development. Imagine a field of grass. A fire has swept through, leaving behind scorched earth. New grass may grow, but the soil itself is changed. This is similar to what happens in field cancerization. The surrounding tissue may appear healthy, but the microbiome has been altered, making it more susceptible to cancer. Field cancerization has profound implications for cancer prevention and treatment. It suggests that monitoring the microbiome of high-risk individuals could help identify those who are more likely to develop oral squamous cell carcinoma. Furthermore, targeting the microbiome in the surrounding tissue could potentially prevent or delay the development of cancer. Section 5. Cutibacterium acnes, a microbial key to unlocking cancer's secrets. Among the many microbes present in the oral microbiome, one stood out in the study Cutibacterium acnes. This bacterium, typically associated with acne, was found to play a significant role in OSCC. The study revealed that Cutibacterium acnes can directly activate the MIC oncogene in oral cancer cells. The MIC oncogene is like a switch that, when flipped, can send cells down a path of uncontrolled growth, a hallmark of cancer. Cutibacterium acnes in this case is the finger that flips the switch. This finding highlights the intricate link between specific microbes and cancer development. This discovery could pave the way for new targeted therapies. By understanding how Cutibacterium acnes interacts with cancer cells, scientists may be able to develop drugs that block this interaction and inhibit tumor growth. This finding underscores the importance of investigating the roles of individual microbial species in cancer.
Dr. Valentino, to the OR, please.
Section 6. A Universe Within, Exploring the Multi-Kingdom Microbiome. This study delved deeper into the microbiome, revealing a complex ecosystem with multiple players. It's not just about bacteria. Fungi, viruses, and even bacteriophages play a role. Bacteriophages, viruses that infect bacteria, are particularly intriguing. They can influence the composition and function of the bacterial community, indirectly impacting OSCC development. Imagine a vast cosmic web, with each node representing a different microorganism. Their interactions create a delicate balance. Disrupting this balance, even in a seemingly insignificant way, can have cascading effects. Understanding these intricate relationships is crucial for developing effective microbiome-based therapies. The multi-kingdom nature of the microbiome adds another layer of complexity to OSCC research. Future studies need to consider all members of this intricate community and their interactions to fully understand their roles in cancer development. Section 7. Navigating the cosmos of research, acknowledging the study's limitations. While this study provides groundbreaking insights, it is important to acknowledge its limitations. The sample size was relatively small, and the study focused primarily on one specific subsite of oral cancer. Future research with larger and more diverse patient populations is needed to confirm these findings and explore their applicability to other subtypes of OSCCC. Scientific exploration is like navigating a vast, uncharted cosmos. Every study, a journey of discovery. We must acknowledge the limitations of each expedition. This study provides a crucial first step, a beacon guiding us towards a deeper understanding of the microbiome's role in OSCC. Further exploration is necessary to fully unravel its complexities. Acknowledging these limitations does not diminish the significance of this research. It highlights the need for continued investigation. Each study builds upon the last, gradually illuminating the path towards more effective treatments for OSCC. Section 8, Towards a New Frontier Microbiome-Targeted Therapies. The implications of this study extend far beyond the laboratory. It paves the way for a new frontier in cancer treatment microbiome-targeted therapies. Imagine a future where we can manipulate the microbiome to fight cancer. This could involve using probiotics to introduce beneficial bacteria, prebiotics to nourish them, or even fecal microbiota transplantation to replace a diseased microbiome with a healthy one. This is not science fiction. These are real possibilities within our reach. By harnessing the power of the microbiome, we can develop personalized treatment strategies tailored to each patient's unique microbial profile. The development of microbiome-targeted therapies is still in its infancy. However, the potential benefits are immense. These therapies offer a less invasive and potentially more effective approach to treating OSCC. This is an exciting time for cancer research, and the future holds great promise. Section 9. A final word, the future of oral cancer treatment, a symphony of collaboration. The study of the microbiome is a complex and rapidly evolving field. Unraveling the intricacies of its role in OSCC requires a symphony of collaboration. Scientists, clinicians, and patients must work together to translate these findings into tangible benefits for those affected by this disease. This research represents a paradigm shift in our understanding of OSCC. It highlights the importance of considering the microbiome in all aspects of cancer care, from prevention and diagnosis to treatment and survivorship. The future of oral cancer treatment lies in embracing this new frontier and working together to harness its full potential. The journey ahead may be long and challenging. However, the potential rewards are immeasurable. By working together, we can unlock the secrets of the microbiome and pave the way for a brighter future for those affected by OSCC.